In this lab, we're going to be discussing E. coli and coliforms. Step 1 of the lab is to make a 1 to 10 dilution. After combining the 10 grams of your sample and 90 mLs of buffered peptone water, make dilutions. Step 2 is to spread plate dilutions on VRB agar. To do this, you're going to spread 100 microliters onto VRB plates in duplicate. Step 3 is to conduct most probable number testing using LST or lauryl sulfate tryptose tubes. Add 1 ml from each dilution into each LST tube. Make sure that this is done in triplicate as we are using a 3-tube MPN scheme. Step 4 is to plate your dilutions onto E. coli Petri films. To do this, you're going to add 1 ml of each dilution onto Petri films. Just like the plating, you're going to want to do the Petri films in duplicate. Add the spreader with the ridge side down onto the Petri film after plating each dilution. Gently press each spreader to ensure even distribution. Step 5 is to inoculate your biochemical test tubes. Add 1 loopful of pure culture to each tube. Tubes include tryptone broth, MRVP broth, and Koester citrate broth. Step 6 is to incubate your VRB plates, Petri films, and biochemical tubes at 37 degrees Celsius. 24 hours later. Step 7 is to count your VRB plates. On VRB plates, E. coli colonies are red to pink with a red precipitate around the edge of the colony. This is due to the fact that there is a pH indicator in VRB agar called neutral red, and the fact that the fermentable sugar in VRB is lactose. Bacteria like E. coli that can ferment lactose will produce these red to pink colonies, while bacteria like Salmonella that cannot ferment lactose will appear colorless. Here's just another view of the bottom of a plate. The next step is to streak our eosin methylene blue or EMB plates. Using an isolated colony from your VRB plate, streak an EMB plate. Incubate your plates at 37 degrees Celsius for 24 hours. Step 9 is to count your Petri films. Count your blue or red colonies separately that have gas production. Colonies that are red are considered presumed coliforms. Colonies that are blue are considered confirmed E. coli. Review the 3M interpretation guidelines in Blackboard for help with how to count colonies on Petri films. 24 more hours later. Step 10 is to check for your positive LST tubes. Positive LST tubes have turbidity and gas production in the Durham tube, like the tube on the left. Negative LST tubes will have no gas production and/or no turbidity. Step 11 is to inoculate BGLB or brilliant green lactose bile and EC or E. coli broth MPN tubes. To do this, you're going to transfer 1 ml of each positive LST tube into a BGLB and an E. coli broth tube. Once this is done for all positive tubes, incubate your BGLB broth tubes at 37 degrees Celsius for 48 hours. E. coli broth tubes will be inoculated at 44.5 degrees Celsius for 48 hours. Step 12 is to check your EMB streak plates. E. coli colonies on EMB plates will be blue or black with a metallic green sheen as shown in this picture. Come back on Wednesday for videos on how to conduct the biochemical tests.